Hello students. So this is enhancement of food production, part 9. So we have discussed the matter which was related, the steps which were related to the sewage treatment. There were various steps which were involved in the sewage treatment, that is recycling of the sewage, that we have studied in detail in the last session. So today we are going to discuss about the microbes in the energy generation. So many developing countries are encouraging for installation of biogas plants to meet out the requirement of energy. So why there is need of biogas plants? Because of the energy production. The more energy is required. So biogas is used as domestic as well as industrial fuel. It is non-conventional and renewable energy source and is obtained by microbial fermentation. This is important part, and because of that the biogas plants are more demand. Okay, so biogas is a mixture of methane, number of gases they are involved in this. Okay, so these are the number of gases which are very important, which you have to remember. So biogas is a mixture of methane, carbon dioxide, H2S, and other gases in traces in small amount. Biogas is highly flammable and is used as a source of energy. This is very important point. Plant wastes, animal wastes, and domestic wastes, as well as agricultural wastes, municipal wastes, and forestry wastes, etc., are commonly used for the biogas production. So, from the waste, we are getting biogas. That is very important part of this. Why biogas is in demand? Because we are getting best out of waste. Okay, so these are the number of waste products which are produced because from the plants or animals, domestic waste, agriculture, municipal, forestry, and etc. So because of all the wastes we get, there is no use of the such waste products, right? We throw them out, so all such wastes can be used in the biogas production. So cattle dung is most commonly employed substrate for biogas production, and it is rich source of cellulose from plants. Again, this is very important part of this. So biogas production, how it is carried out. Most commonly used models of biogas plants are KVIC and IARI. These are the two models which are used for the production of biogas. The digester used for biogas production is called biogas plant. So, what is a biogas plant? The utensil or digester which is used for the biogas production, that plant is called as biogas plant. A typical biogas plant using cattle dung as a raw material consists of a digester and gas holder. Digester is made up of concrete bricks and cement or steel. So these are the different parts. So what the biogas plant consists of? It consists of two parts: a digester and a gas holder. This is important. Out of these two, digester is made up of concrete bricks and cement or steel. Now this is important part. Okay, so there is cylindrical gas holder or gas tank above it to collect the gases. So cylindrical gas, what is the use of this holder? It collects all the gases which are formed naturally in that cylinder, okay, in that digester. So digester has a side opening that is called as charge pit. So that is a side opening which is present and which is attached to the digester. The cow dung is fed from which the digester is partly buried in the soil. Anaerobic digestion involves in three processes. So it is not completely buried inside the soil and it is not completely above the soil, but it is partly inside the soil and partly above the ground. Okay, so these are some steps like hydrolysis and solubilization. So before going to study that, I think this part is important. Let's try to understand the biogas plant, how it looks like. So this is the charge pit. Okay, this is the opening. Dung water is, now this is the input. Okay, and this is a pipe you can see, which is attached to this digester. This is the gas holder. Now digester, in this the raw material is or all the wastes is they are suspended in this part, and because of the natural digestion or whatever chemical reactions they occur inside this digester, because of all this digestion or of the wastes, all the gases they get evaporated, okay, they get collected. So that those collected gases they are collected in this gas holder, okay, and from this gas holder you can say this is a pipeline. This is called as biogas. Now this all the mixture of gases which are produced from these digestion of these wastes, now all the gases they are collected through this pipe. This is sludge, okay, and this is a biogas plant, how it looks like. Now let's have a look how a biogas plant it looks. So you can observe in this diagram, see how it is partly buried, you can observe in this, right? So it is some part is buried inside the soil and some part is above the soil, okay. So the part which we cannot see, which is inside the soil, that is the digester part, where the natural mechanism is occurring, the all the wastes they are getting digested in that tank. Now you can observe this another pit like structure, that another structure is nothing but from which the input is added, okay, all the waste material is added from this, and somehow this there is a pipe which is attached to this digester. So from this through that pipe all the raw material gets collected inside this digester, and all the gases they are collected in this part, okay, and through which now in this gas tank you can say, and from this you can see this is a pipe. Through this pipe all the gases they are collected, okay. Now this pipe contains the biogas, and this biogas is collected, okay. So that's all about this biogas plant. So you can observe now this is inlet small scale biogas plant. All the slurry is situated in this digesting tank. This is a biogas reactor. Biogas get formed through this pipe. The biogas gets collected, and excess digested whatever material is, it is the outlet for that, okay. So okay, so I think that is much more clear now to you, that how a biogas plant it looks like and how it works. So let's have a look that what are the steps how the anaerobic digestion occurs inside the biogas plant. So first step is hydrolysis or solubilization. In initial stage, the raw material, or that is called as cattle dung, is mixed with water in equal proportion, and that's why it is called as slurry, okay. That is called as slurry. So the cattle dung is mixed with the water in equal proportion. So there is equal proportion of that raw material, whatever we are adding, all the wastes which we are adding in that. For example, we are adding cattle dung. So equal amount of water is added into that part, and it becomes a slurry, and then it is fed into the digester. Then it is added inside the digester. Here, anaerobic hydrolytic bacteria. These are the names of bacteria. Now these are the names which you have to remember while writing the answer. So these they hydrolyze the carbohydrates into simple sugar. So in the hydrolysis, this is the first step where we are talking about the hydrolysis or solubilization, okay. So what is getting hydrolyzed? The cattle dung is getting hydrolyzed, that is the raw material which is in the form of slurry, okay. And it is getting hydrolyzed into simple sugars and proteins, or into amino acids and lipids, okay. And which through which bacteria? These are the anaerobic hydrolytic bacteria. Their names are Clostridium and Pseudomonas, okay. So this is the first step of hydrolysis or solubilization. Second step is the acidogenesis. In the second step, the facultative anaerobic or acidogenic bacteria or you can say the obligate anaerobic organisms. They convert this simple organic material into acids like formic acids, acetic acid, and hydrogen and carbon dioxide. So in the acidogenesis, in short, what happens because of the activities of facultative anaerobic acidogenic bacteria or obligate anaerobic organisms which are present inside that material or raw material. So because of the presence of these bacteria or because of the presence of those prokaryotic cells, there is a process that is called as acidogenesis which occurs. And because of the acidogenesis, all whatever is getting converted is in the form of acids like formic acid, acetic acids, and hydrogen carbon dioxide gases they get formed. Third step is called as methanogenesis. Now this is very important as far as the hydrolysis or as far as the biogas plant is concerned. This is last stage in which the anaerobic, now this is the last step of this biogas plant. Okay, so this is again very important, in which the anaerobic methanogenic, now the important thing is methanogenic anaerobic bacteria, they play very important role in this methanogenesis. So these are the methanobacterium or methanococcus. These are the names of those microorganisms which convert the acetate. Now acetate is getting formed in the earlier step, so they convert the acetate hydrogen carbon dioxide into methane, carbon dioxide and water, and the other products. So you can observe these are the chemical reactions through which whatever is formed in the earlier stage in the acidogenesis, that's get converted into methane. Okay, so what are the benefits? Now you can get a question on this, as this lesson is having less weightage, so you can get a question on what are the benefits of biogas plant. So you have to remember all the points. That's the biogas. What are the benefits? That it is a cheap, safe, and renewable source of energy. Okay, so it is cheap, it is safe, it is a renewable source of energy. It can be easily generated, stored, and transported. So this is the first and very important benefit of this. Second important benefit of it is it can be used for domestic lighting, cooking, street lighting, as well as small scale industries. So these are the applications of biogas, where biogases can be used in the domestic lighting, they can be used in the domestic cooking, street lighting, as well as in the small scale industries also. These biogases they can be used. It burns with blue flame without smoke, again very important. So it is a healthy gas, you can say. It is not harmful to the environment, as it burns in blue flame, and again there is no smoke. So this is again very important benefit of this biogas. It helps to improve the sanitation of the surrounding. It is eco-friendly and does not cause pollution and imbalance of the environment. So it keeps the environment in balance. It is eco-friendly, and it does not cause any pollution. uh, Sludge which is leftover is used as fertilizer. So whatever is leftover, or we have studied that whatever is wastes, okay, so all that wastes is getting collected and it is added to produce this biogas. So all this leftover is used as a fertilizer. So these are the basic benefits, you can say, of the biogas plant. Now role of microbes as biocontrol agents. So what part you have to remember from this lesson is number of very short answer questions, number of MCQs, and as far as your need is concerned, for that the names of microorganisms are very important. So even if the weightage of this lesson is very less, but the names of the detailings with whatever detailing is used in this lesson, so that part is very important. Okay, so don't miss that part. So let's have a look at what is the role of microbes as biocontrol agents now. The term biocontrol refers to the use of biological methods to control diseases and pests. So here we are talking about the biocontrol agents. Okay, so these are the biocontrolling, how they control the biological pests, or how they control the diseases and the pests. Okay, so that is nothing but the biocontrol mechanism. So, the natural method of eliminating and controlling insects, pests, and other disease causing agents is by using the natural and biological enemies. This is called as biocontrol, uh, or biological control. So, by using the natural or biological methods, the insects, pests, and other disease causing agents, they can be controlled. So that type of control is called as biocontrol or biological control. The agents which are employed for this are called as biocontrol agents. So here we are talking about the microorganisms and how they are used as the biocontrol agents, or how they play a very important role as biocontrol agents. Okay, so microbes are one among them. These microbes include bacteria. Now, which are the microbes? They can be used as biocontrol agents, not only the bacteria but also some fungi, some viruses, some protozoa. They are also used as biocontrolling agents. Microbes as biocontrol agents act in three ways: either they cause the disease to the pest, or compete or kill them. Chemicals, insecticides, and pesticides are extremely harmful to human beings, as pollute the environment. So they imbalance the environment, in short. Okay, so if we use lots amount of chemicals, insecticide, pesticides, so they are extremely harmful to the human beings, uh, or they pollute the environment. Okay, so avoid that. So to avoid the use of such chemicals, insecticides, and pesticides, you can say the biocontrol agents or biocontrol measures will greatly reduce our dependence on toxic chemicals and pesticides. So to avoid that, we can use the biocontrol agents. So these are the examples of biocontrol agents. So see how the Bacillus thuringiensis. Now this is the name of a microorganism. This name you have to remember. So Bacillus thuringiensis, that is called as BT, is used to get rid of butterfly caterpillars where dried spores of Bacillus thuringiensis are mixed with water. Now see what we are getting rid of. We want to get rid of these butterflies, we want to get rid of the caterpillars. So we don't want the butterflies and caterpillars. So if we don't use the biocontrol agents, and in that case we can avoid the butterflies and caterpillars, or we can kill them with the help of these toxic chemicals or toxic pesticides. Okay, so in the biocontrol agent, microbial biocontrolling mechanism, what we do is we dry the spores of Bacillus thuringiensis. Okay, so the spores of BT or Bacillus thuringiensis, they are taken. This is the first step. In the next step, all these spores, they are mixed with water and they are spread onto the vulnerable plants such as brassicas and fruit trees. So all that water is sprayed on that, or on those plants or trees. These spores, what they do now, they are then eaten by the insect larvae. These spores, which are sprayed on the trees or on the plants, they get eaten by the insect larvae. So, as the insect larvae they eat such spores, in the gut of the larvae, the toxin that is called as cryprotein is get released, and the larvae get killed eventually. Again, what we are getting, we are killing the larvae, but not with the help of the chemical pesticides or insecticides, but by using the biocontrol mechanism. We are using the spores, we are mixing the spores in water, we are spraying those spores on the plants and trees, and as the insect larvae they eat those spores, so in the gut region the cry protein get released, the toxin which is present in the cry protein, it's get released, and because of that toxin the larvae get killed eventually. Okay, next is the Trichoderma species. These are the free living fungi found on the root ecosystem or rhizosphere. So these are the Trichoderma. How they are used? They are effective as biocontrol agents of several soil-borne fungal plant pathogens. The fungus produces substances like viridin, glyotoxin, glyoviridin, okay, glyovirin, etc. So that inhibit the other soil-borne pathogens attacking roots, rhizomes, etc., causing the rot disease. So they get controlled by this Trichoderma species. So four groups of biocontrol agents are known. They are bacteria, fungi, viruses, and protozoa. Microbial pesticides and their host. So the correlation is depicted as in the following table. So this is very important table where pathogen is Bt, that is Bacillus thuringiensis, and what is the host? Caterpillars, cabbage worm, adult beetle. Okay, fungi. So this is the table where we talk about, for example, nucleo polyhedrovirus, that is NPV. So these viruses, these are the pathogens, and what is the host? Caterpillars. We can control the caterpillars, moth, ants, wasps, and beetles by using these viruses. Okay, so bioherbicides, now what are they? Weeds are unwanted plants that grow in agricultural fields, ponds, lakes, etc. So these weeds compete with the main crop in the farmland for water, space, minerals, light, air, etc. and also act as collateral hosts for several pathogens. So we don't want such weeds. So to remove that, microbes are also used as herbicides. Many dicotypes that grow in the field of cereals as well as weeds can be killed by certain microbes. Now, these are the microbial herbicides and their host. So, these are the pathogenic fungi as mycoherbicides. These are the names like Phytophthora, they control the milkweeds, Alternaria crassa, control water, hyacinth, then Fusarium species, it controls most of the weeds. Bacterial pathogens, they act as herbicides like Pseudomonas species, it attacks several weeds. Xanthomonas, it attacks several weeds, and Agrobacterium, which attacks the several weeds. Now, this part is important as you can see each and every microorganism, that pathogen is different and the host is different. Okay, so these names are important. This is the part of the earlier part. Okay, so that part is important. Now, role of microbes as biofertilizers. Biofertilizers, see here we are talking about the biofertilizers, not the chemical fertilizers. So fertilizers are nutrients which are necessary for the growth of plants. We all know fertilizers, they are used for the growth of better growth of the plant and thus for the productivity of cultivated plants. Use of fertilizer for increasing productivity is one of the aspects of green revolution. So by using these biofertilizers, we can get lots of production of the plant. So the productivity of the plants can get increased by using the fertilizers. So these fertilizers, they are again classified into two: inorganic, that is chemical, which is where the chemicals they are used or they are added. Another is biological, they are also known as organic. So inorganic fertilizers, they are synthetic, they are artificially made, where mineral salts of NPK are mixed in definite proportion and then dusted in the fields. Okay, so these are the inorganic fertilizers. While non-judicious or excessive use of such fertilizers lead to pollution of soil, if we use such type of fertilizers which are synthetic or which are artificially produced, that there are lots of disadvantages, like excessive use of such fertilizers lead to pollution of soil, air, and groundwater. This is a long term of such inorganic fertilizers can damage the soil, the air, and the groundwater, and the soil can become acidic. Again, it is the biggest disadvantages, one of the disadvantages of the inorganic biofertilizers. So organic fertilizers are biological in origin, and they include farmyard manure, compost, green manure. Okay, use of these fertilizers increase the fertility of soil. So these are comparatively uh, healthy to use. Okay, comparatively to the inorganic or synthetic fertilizers. Nowadays, for better and sustainable agricultural production, farmers use biofertilizers. So, for sustainable, better agricultural production, the biofertilizers, they are uh, used in a large amount. So biofertilizers are mostly nitrogen fixing. They fix the nitrogen which is present in the environment, living microorganisms which enrich the nutrient quality of the soil. They include bacteria, cyanobacteria, and fungi. So lots of species of cyanobacteria, lots of species of bacteria, fungi. They are used. These are the microorganisms. They are added in the biofertilizers, and because of the use of such microorganisms, the soil becomes very enriched. Okay, so biofertilizers are commercial preparation of ready to use live bacterial or fungal formulations. Their application to plant and soil or composting pits helps to enrich. Now these are some of the advantages of the biofertilizers. These are very important. They can be asked for the short note. So as we are using live bacteria or fungal formulations in that, the application to plant soil or composting pits helps to enrich the soil fertility. They enrich the soil fertility due to their biological activity, because of their natural biological activities. Use of biofertilizers is cost effective and eco-friendly. This is very very important. There are no any side effects on the environment. They are very eco-friendly. They play a vital role. They play a very important role in maintaining a long-term soil fertility and sustainability. So types of biofertilizers. Now on the basis of nature and function of biofertilizers, they are divided into some groups like nitrogen fixing biofertilizers. So these are the nitrogen fixing microorganisms which which can convert the atmospheric nitrogen into nitrogenous compounds like nitrites, nitrates via ammonia. So the conversion is possible because of these nitrogen fixing biofertilizers. So these microorganisms, they are called as diazotropes, and they are of two types: symbiotic nitrogen fixing microorganisms like Rhizobium and Arabina. These are the example. So they are present on the underground parts, that is roots of the higher plants, that we are going to see with the help of diagrams. Free living and non-symbiotic like Azotobacter, which are non-symbiotic or they are free living. Okay, Clostridium, Azotobacter, Nostoc, these are the examples. Phosphate solubilizing biofertilizers. These are bacterial species which solubilize the insoluble inorganic phosphate compounds, such as rock phosphate. So again, this is another kind of or another group of biofertilizers. For example, Pseudomonas species, they are used in that. Aspergillus species, they are used in that. Making biofertilizer. Composting is a natural process that turns organic material into dark, rich substance that is called as humus or compost, which is again very useful for the enrichment of soil. So the composting process is dependent on microorganisms to break down the organic matter into compost. There are many types of microorganisms found in active compost, and these are bacteria, fungi, actinobacteria, protozoa, and rotifers. So these are the various kinds of microorganisms. Cyanobacteria as biofertilizers. The many cyanobacteria are aquatic and terrestrial, free living. They are symbiotic, some are aerobic, some are photosynthetic, nitrogen fixing, heterocystis or non heterocystis forms, anabina, non-stock, uh, oscillatoria. These are the examples. Okay, they are they are associated with the lichens. Anabina is associated with the plants like azolla, cycas, etc. So let's have a look like fungal biofertilizers. Mycorrhiza is a fungus. It forms symbiotic association with the underground parts. Symbiotic association, that means both of the plants or both of the members, they get advantages from each other. Okay, so one example, these are of two types: ectomycorrhizae. They have well developed mycelium that form mantle on the outside of the root. Let's have a look in the diagram how they look. So ectobacterium. Sure. Now these are the ectobacterium. You can observe in this diagram. Again, uh, another example is endobacterium. You can observe another diagram. This is again another diagram. You can observe that how the ectomycorrhiza they look like. Okay, you can observe in this structure now. See, ectomycorrhizae. Now this is xylem. This is epidermis, outermost part. Cortex is present here inside. This is hearting net. This is fungal sheath. Now this is the fungal sheath. Okay. Ecto means outside. You can observe outside of that epidermis. Now this is the epidermis. To the outer part of this epidermis, somehow this ectomycorrhizae, they are their sheath. You can see it is observable. Okay. So ectomycorrhizae, they have well developed mycelium. You have observed in the diagram. They are present outside of the roots. They are forming a layer. This increases the absorptive surface area of roots and accelerates uptake of water and nutrients. So because of this, it increases the absorptive surface area of the roots and accelerates the uptake of water, nutrients. Okay, so that is again very important part. Due to this plant, the plant vigor, growth, and yield of the plant, it gets increased because of the presence of these endomycorrhiza, you can say. Sorry, ectomycorrhiza. Some hyphae of the mycorrhizal fungus penetrate into the root. They form hectic net in the intercellular spaces of the root cortex. Now, endomycorrhizae, they grow in between within the cortical cells. Now, here, endo, there is a difference between the two terms, endo and ecto. Ecto means outside, endo means inside. So they are within the cortical cells, they are present in between or within the cortical cells. Fungal hyphae penetrate in the cells and form finely branched orbicles intracellularly, and we form vesicles mostly in the intracellular spaces of the cortical cells. So you can observe in the diagram how beautifully it is shown, as we have observed just the diagram. Endomycorrhizae, see, they are present inside the cells. You can observe in the diagram where where they are situated. Okay, so they are present inside these cells. Endomycorrhizae. Okay, so they are called as vesiculo or vascular mycorrhizae. That is called as VAM. So you can get a short note on that. Write a short note on VAM or write a short note on vesiculo or vascular mycorrhizae. So nowadays they are described as AM fungi. The plants with VM grow luxuriantly in less irrigated lands. The association of VAM with crop plants helps in the conversion of less productive field into more productive field. Again, this is very important part of this. Okay, that part you have to remember. So you can get a short note on this, or you can get a differentiate between ectomycorrhizae and endomycorrhizae like of question. Now, biofertilizer as microorganisms. Rhizobium, they are rod shaped, motile, they are aerobic, gram negative, spore forming, nitrogen fixing bacteria. They contain NOD genes or NIF genes. Okay, this is important part. These are the characteristics of the rhizobium. So you can observe in the diagram. You can observe in the diagram that how the rhizobium it looks like. Yes, so this is the rhizobium. You can observe in this diagram, these are the root nodules. Okay, and Rhizobium is situated. You can observe in this diagram. Like now, see, this is the symbiotic association. Rhizobium bacteria, it has the ability to take atmospheric nitrogen and convert it into soluble form. Now, Rhizobium, it acts as the symbiont. So you can observe this is the soil, and the roots, they are in beneath, they are inside the soil, and to these root nodules, they are formed, and these are the Rhizobium. Okay, so you can observe in such diagrams, these are the tumor-like structures. Okay, where you can observe, like, see, these are the Rhizobium. Okay, so they contain the Nod gene and Nif genes. They are rod-shaped bacilli. They are motile. They can move, aerobic, without they need oxygen. Gram negative. When they are stained, they are pink in color. They are non-spore forming. They do not form spore-like structures. They are nitrogen fixing bacteria, and they contain Nod genes and Nif genes. They form symbiotic association with the roots of leguminous plants. So, as I have shown you just the diagram, so they are associated with those plants. They bring about nodule formation on the roots, multiply inside the nodule. So, inside the nodules, they get multiplied. So, inside this part, they get multiplied. Okay, they get multiplied in this part. That is, you can say, that is the shelter of such microorganisms. What is their role? They fix atmospheric nitrogen into organic forms, which can be used in the plants as nutrients. Okay, so this is again very important part. Azotobacter, now it is the important and cell and it is well known free living. It is free living, nitrogen fixing, aerobic, non photosynthetic, and non nodule forming bacteria. It is intimately associated with the roots of grasses of certain plants. Now, these azotobacter, they don't form any kind of nodules. Okay, they don't form any kind of nodules, but they form just they are just attached to the uh, parts of the plants. That is, they are just intimately associated with the uh, parts of the plants. So these are the azotobacter. In this diagram, you can observe how the azotobacter they look like. Yes, these are nitrogen fixing microbes. You can observe in this diagram. They are pink in color again, but they are not. They are in now. You if you observe this plant, they are not forming any root nodules. Okay, they just they just present on the surface of the roots. Okay, so they just uh, cover the surface area of the roots of the plants. Okay, and these are the bacterial cells, azotobacter. You can observe these are the various kinds of diagram of such type of microorganism. Rhizobium, azotobacterium. Okay, so it is used as biofertilizer for all non-leguminous plants, especially rice, cotton, vegetables, etc. Now, Azospirillum, it is free-living, aerobic, nitrogen-fixing bacterium associated with the roots of corn, wheat, and jawar. Now, again, the plants they are specified: wheat and jawar, corn. These are the roots where this Azospirillum is situated or associated. It fixes the considerable quantity of nitrogen and non-leguminous plants such as cereals, millets, cotton, oilseeds, etc. Represent. Now let's have a look how they look like. So Azospirillum. So in this diagram you can observe the Azospirillum, which we are using as the biofertilizer. This is the biofertilizer. These are the Azospirillum cells which are used as the biofertilizers. Okay, so they also do the biofertilizer fixation. Okay, nitrogen fixation. Such type of mechanisms they are carried out. Okay, Anabina. Now it is a genus of multicellular. Now it is multicellular filamentous cyanobacteria that exists as plankton. Let's have a look what is a filamentous cyanobacterial cell. Now this is Anabina. Yeah, these are the cells. Okay, so they are Anabina, multicellular. You can observe many cells they are attached, right? They are in the form of chain. You can see heterocyst and so these are the various kinds of structures you can observe in this granulation which is present in the cells. So these are the many cells. That's why it is not single cellular but it is multicellular. Again it is filamentous. It forms a filamentous like structure, so it forms a filament, a chain like structure. Okay, you can observe in this, in this diagram how it looks. These are the filamentous structures. Okay, and another part is they have the ability to fix the nitrogen, and they also form symbiotic relationship with certain plants like caroloid roots of cycas or anthocyrus thallus. It has some specialized and colorless cells. They are called as heterocysts. These are the sites of nitrogen fixation. So as we have studied in the, now these you can observe in this, these are the heterocysts. Okay, so these are the heterocysts. Heterocysts, they are colorless cell. You can observe the other cells, they are different in color, while the heterocysts, they are not colored. They are colorless. Okay, they are colorless. So that's all about the Anabina. The next is Azolla. Azolla is a free floating water fern. Now it's a free floating fern. Okay, it's another kind of species now. Azolla plant consists of floating rhizome stem with small overlapping bilobed leaves and roots. The leaf shows dorsal and ventral lobes. So you can observe how the azolla they float on the water. Now see, this is the structure of azolla. You can observe that how it floats on the water surface. Now see, it is floating on the water surface. Okay, so this green color part, this is nothing but the azolla which is used as the biofertilizer. Now these are the crops and the biofertilizer is used. Now azolla is a free floating water fern. It's a free floating water fern. It floats on the water surface. And it what type of leaves can you see? These are the leaves are bilobed. Okay, these leaves you can observe in this, the leaves they are bilobed. Okay, and they show dorsal and ventral lobes. In the dorsal lobe, anabina filaments they are present in the parenchyma, which fixes the nitrogen. So parenchyma it fixes the nitrogen. Because of that the nitrogen fixation is possible, as well as can be used as biofertilizer in the field. So this is all about the azolla. You can see how it looks, the leaves of the azolla. It is free floating and it is very uh, helpful in the fixation of nitrogen. Okay, so what are the benefits of biofertilizer now? They low cost, can be used as marginal, but they can be used by marginal farmers. So it is not costly. It is free from pollution hazards, so it is pollution free. This is important. Increase in soil fertility. It increases the soil fertility. It increases the BGA as biofertilizers secrete growth promoting substances, organic acid, proteins, vitamins. So all it can be obtained by this. uh, Azotobacter supply nitrogen and antibiotics in the soil. Biofertilizers they increase the physical chemical properties of soil like texture, structure, pH, water holding capacity of soil by providing nutrients and organic matter. So these are the advantages of the biofertilizers. So you can get a question, as the weightage of the lesson is not that much, it contains only two 3 mark 2 mark questions. Prepare all the MCQs. All the names of microorganisms are very important as far as the details of this lesson are confirmed. Okay, so very large आंसर क्वेश्चन भी अगर नहीं आ रहे हैं इस लेसन से, फिर भी वेरी शॉर्ट आंसर क्वेश्चंस की पॉसिबिलिटी बहुत ज़्यादा है, तो वो सब क्वेश्चंस आप इसमें से कर लें। ये कंप्लीट जो थियोरेटिकल लेसन है वो अभी हमने पूरा कर लिया है। नेम ऑफ माइक्रो ऑर्गेनिजम्स जितने भी हैं वो आप इसमें से कर लीजिए। ओके, so that's all about this lesson, that is enhancement of food production. So that's all for today's session. Thank you.